Another aspect of particularly primate reproduction is that we have invasive placentas. Let's take a look at what that means. Implantation of the embryo is aggressive, it's invasive, and this invasiveness is actually ancient, but it was lost in some lineages. The degree of invasiveness varies among mammalian groups. Invasion is something that should give a fitness advantage early in life because it gives the embryo increased control over nutrition and growth. However, there are trade-offs. Greater embryonic control over maternal blood pressure increases the risk of preeclampsia, and animals with more invasive placentas have greater risk of metastatic cancer. So the fact that placentas are invasive really has a number of rather surprising consequences. One is it shows that the embryo and the mother are to a certain degree in conflict with each other, and the embryo is trying to take more control over the mother than the mother would like to give. And as with many other things that we've seen, every time a system comes in that has a big advantage, that also means that evolution is willing to tolerate a big vulnerability or a big cost in order to get that advantage. And in this case, the diseases of pregnancy and the risk of metastatic cancer are two of the costs or vulnerabilities that are associated with invasive placentas. So the pre-embryo is implanting aggressively. The outer embryonic cells, the trophoblast, actually bore into the uterus by secreting enzymes that dissolve the tissue that protects maternal blood vessels. The endometrium is both enabling and limiting this invasion. So there is an overlap of interest between the embryo and the mother, of course, but there is also some conflict of interest. The trophoblast then becomes part of the placenta where it secretes hormones that can mimic maternal hormones. And these hormones influence maternal blood sugar and the dilation of maternal arteries in the placenta. That helps to regulate the delivery of nutrients to the embryo. The degree of invasion and the ability of embryonic tissue to control nutrient flow depend on placental morphology, and that varies among major groups of mammals. So one of the things we're going to be looking at is variation in placental morphology. Now, for our purposes, the critical feature of placental morphology are the ones that determine how many layers of cell and tissue separate fetal blood from maternal blood, and how intimately fetal and maternal tissue are intertwined over how large a surface area. Prior to placenta formation, there are 6 layers of tissue that separate maternal and fetal blood. During placentation, 1, 2, or 3 of the maternal layers can be lost. And the shape of the placenta can be either diffuse, that means it occupies most of the surface of the embryonic sac. It can be cotyledonary, with multiple discrete attachments. It can be zonary, forming a band around the fetus, or it can be discoid, attaching at one point and being smaller than the fetus. We have discoid placentas. So here are the potential fetal and maternal layers. On the left, we have fetal tissue. On the right, we have maternal tissue. This is a fetal blood vessel. This is a maternal blood vessel. The blood vessel is surrounded by an endothelium. That's layer 1. Then there is connective tissue, and then there are the chorionic epithelial cells. When the zygote or the pre-embryo first implants, this would be the layer of cells lying next to the endometrium. Those are the endometrial epithelial cells. Then there is maternal connective tissue. Then there are maternal endothelial cells surrounding the blood vessel, and then there is the delivery of blood itself. So potentially 3 fetal and 3 maternal layers. The fetal extraembryonic membranes all go into forming the placenta, and both fetal and maternal tissue layers can contribute to the structure of the blood vessels, to connective tissue, and epithelial cells can contribute to the placenta. So placental classification based on cell layers has these names: epithelial chorial, which is cows, pigs, and horses, is the full set of 6 membranes. In endothelial chorial, which would be dogs and cats, one layer of maternal tissue, the epithelial layer in the endometrium, has been lost. However, in hemochorial, which characterizes humans and rodents, the growing chorionic villi erode through the maternal endothelium. They form a hemochorial placenta in which the fetal chorionic epithelium is bathed directly in maternal blood. So here is the fetal epithelium here. It's actually in villi. This is just a simplified cross-section. And the maternal blood is actually right up against it. So it's bathed in maternal blood. So when and why did that evolve? And what are the consequences? Well, here is the evolutionary history of layers laid out in a phylogenetic tree. And what we're looking at are, in black, we have epithelial chorial, in red, endothelial chorial, and in blue, hemochorial. And what we can see is the hemochorial placenta is actually ancient. That's not a derived condition. That is an ancient condition. Interestingly, that condition has been lost in certain groups. So, for example, in things like we don't really know what the condition was in a deeper ancestor than that, but in the marsupials, they have an epithelial chorial placenta. So they have the full 6 layers. And that is also the case in the ungulates. So things like pigs and horses and cows and hippopotamuses, they all have epithelial chorial placentas. Then the endothelial chorial, so the one where one tissue layer has been lost on the maternal side, includes things like elephants and aardvarks and moles, European moles, and foxes and cats and fruit bats, also another set of bats. However, humans retain the ancient condition, so they have a hemochorial placenta. The fewer the layers, the shorter the gestation time. So if we looked at 109 species of all mammal orders, then the fewer the number of tissue layers that separate maternal and fetal blood, the shorter the gestation length for a given maternal body mass. So the shift from the non-invasive epithelial chorial placenta to the intermediately invasive endothelial chorial placenta to the highly invasive hemochorial placenta reduces gestation time by 35 days. It might be more accurate to turn that around and say, since the hemochorial placenta is the ancestral condition, that adding tissue layers increased gestation time by 35 days. This is what some of the macromorphology of placentas looks like. So here we have a diffuse placenta. It's characteristic of horses and pigs. The cotyledonary placenta is characteristic of cows. The zonary placenta, which actually wraps around the amniotic sac, is characteristic of carnivores and elephants, so dogs, cats, seals, bears, and elephants. And the discoid placenta is characteristic of primates and rodents. So we have that kind of placenta, and so does a mouse. Here, the light pink is the amniotic sac, and the dark pink is the placenta. These shapes also have an evolutionary history. It turns out that our discoid placenta, which is here in black, is also ancient and primitive. And the derived forms are things like the diffuse placenta found in pigs, the cotyledonary placenta found in a cow, the bidiscoid placenta, which is found in tree shrews, is 2 sets of discs, and the zonary placenta, which wraps around the amniotic sac, is found in foxes, cats, and then in some of the afrotherians, the elephant, the aardvark, the rock hyrax, and so forth. However, our discoid placenta is ancient and primitive. However, what about villi, the internal morphology of the placenta? So it can be folded, lamellar, villous, trabecular, or labyrinthine. The villus part of our placenta, which you can see sketched here. So this is where the actual interface is, and the fetal epithelium is bathed here in maternal blood. So with an extensive surface area, which is created by making a tree-like villus that is protruding into that space, that is derived in humans, okay? And it's also shared with some of the carnivores. So if we take all of the differences in human placentation and we compare them with non-human primates and other placental mammals, what we see is that primary interstitial implantation is something that only occurs in great apes among primates, and if we look among other placental animals, we can find it in guinea pigs and a few other rodents. The other thing here in this table I would like to emphasize is that have to do with deep trophoblast invasion that reaches the inner third of the myometrium. So this is something shared only with great apes among primates, and if we look among other mammals, we can see it with even deeper invasion in some bats and in the guinea pig. So what that means is that the great apes share the risk of preeclampsia with us because this deep invasion seems to be a morphological precondition for that risk, and most rodents and armadillos do not. Now, what about vulnerability? Well, there are striking similarities between invasive placental cells and invasive cancer cells. The cellular mechanisms that stem cells use to invade the placenta may be reused by cancer cells to invade and spread during metastasis. This involves cell adhesion molecules, the extracellular matrix, and metalloproteinases that are in the context of extracellular matrix. Angiogenesis is also a feature of both implantation and cancer spread. We now know at least 11 genes that are expressed both in normal trophoblast tissue and in malignant tumors. So understanding the maternal mechanisms that control trophoblast invasion may help us to improve methods of controlling metastasis. A comparative study showed that animals that have less invasive placentation have less risk of metastatic cancer. So here we have 4 panels for 4 different kinds of cancer. The bars on the x-axis are basically cows, horses, cats, and dogs. The x-axis is percentage of malignant tumors, and this is for skin cancer, glandular epithelium, non-glandular epithelium, and connective tissue. And the pattern that we see basically is that for 3 out of 4 of these cancer types, skin, glandular epithelium, and connective tissue, the risk of metastatic cancer is much higher in the groups that have invasive placentation and much lower in those that do not have invasive placentation. So cows and horses are at less risk of metastatic cancer than are dogs and cats. To summarize, mammalian placentas vary widely in the number of tissue layers that separate the fetal and maternal blood, in the shape of the structures that mediate the exchange, and in the degree of fetal invasion of maternal tissue. Human placentas are a mixture of ancient and derived features. Deep invasion is derived. We share it with chimpanzees and gorillas, and it is associated with the risk of preeclampsia, of high maternal, dangerously high maternal blood pressure during pregnancy. Trophoblast invasion pre-adapts cells for metastasis. It is an early life fitness benefit that's trading off with a late life risk. So it's a very good example of antagonistic pleiotropy. Animals that have less invasive placentation have less risk of metastatic cancer.